Hi, I'm Priyal D'Souza. Welcome to my talk. Today I'm going to continue talking about clinical genomics, and today's topic is PCR in clinical diagnostics. I defined clinical genomics in my last presentation, and I hope you remember that. I previously described common types of genomics analysis, and today I'm going to be talking about PCR, which is related to gene amplification based um, testing, which has come on board since the late 1980s. Of course, in my previous talk, I talked about target enriched NGS panels. So what is PCR? PCR stands for polymerase chain reaction, and it's a biochemical analytical procedure which uses thermostable enzymes. In other words, enzymes which are work under extremely high temperatures. It's a process that was invented by Kerry Mullis in 1988, and it's such an important process that he won the Nobel Prize in Chemistry in 1933. Um, the The process enables detection as well as measurement of the copy number, so it can detect either single point mutations, deletions, insertions, as well as a gene, a presence or absence of a gene. It can also measure the copy number, as it's a quantitative process as well as being a qualitative one. Well, how does it do it? Well, it does it by amplifying a few copies more than 1,000,000 times, and it enables to see a DNA or an RNA molecule which is otherwise undetectable. There are two types of PCR, and I'm going to talk about their applications. There's endpoint PCR, which is largely used in R&D labs, and the more clinically oriented one is quantitative or qPCR, also known as real-time PCR. The applications that each of these are used in is for endpoint PCR or RT-PCR, where you can actually have an RNA molecule as your template. It includes single and multiple locus mutation tests, things like RFLP, restriction fragment length polymorphism, sequencing, single-stranded conformation polymorphism, cloning, and gene synthesis. For quantitative PCR or RT-PCR, you can use it for genotyping, mutation detection, copy number variation, and also for pathogen detection, and especially in viral detection, like for coronavirus. So what is needed to perform a qPCR or a qRT-PCR? First of all, you need a template, which is a DNA or an RNA sample from a patient. And you need to carry it out in either a microcentrifuge tube or microtiter well. You need a thermostable DNA polymerase such as TAC, which is a polymerase isolated from a thermophile called Thermus aquaticus. For RNA template, you need to have a reverse transcriptase added as well. You need to have the building blocks, which are deoxynucleotide triphosphates, and two DNA primers, which are prerequisite for the enzymes to work and build new DNA. Each of the primers will bind to separate ends of the amplicon on either strand. You need an enzyme buffer, and you need chemical ions, and the chemical ions which are important are potassium, which helps to keep the DNA strands rigid and separate, a bit like spaghetti when you add salt to your pasta to keep it from sticking together. And this magnesium ions are important as a cofactor for the polymerase. You also need a probe primer if you want very specific detection of the assay, and these are already pre-designed in the kits you buy. Otherwise, you can use cyber green, which is a specific stain for DNA, which will light up the reaction and fluoresce during the process, and causing it to be measured on the PCR machine. Finally, you need the PCR instrument, which will carry out all the measurement of the process of the progress of the reaction.